Thank you, Malte. Hello, everyone. I'm very grateful to have the opportunity to talk to you today and show you our data. My title also is a bit changed than the original one. So I'm going to talk to you about engineering a neurocardiac interface. And I heard a lot of talks the last 2 days, which were really inspiring. And of course, they are all, or a lot, have to do with cardiac regeneration. And we are interested in electrically excitable cell networks, but not only of the heart, but also of the brain. And although the two organs are really different, they do share some aspects. For example, they both really are depending on electrically excitable cells. So neurons and cardiomyocytes. And these cells, of course, are regulated by channels. And it is important to remember that if you have a channelopathy, so if you have a mutation in a specific channel, it can be also common, and therefore it would lead to a dysregulation in the brain in the form of an epilepsy and in the heart in the form of an arrhythmia. And last but not least, of course, we know that both of these organs have a very low regenerative potential. And we also know that they are very important for life and that they have to communicate via the autonomic nervous system. We know that the brain, via the sympathetic and the parasympathetic neurons, will really dictate the contractility and the heart rate. And we know that we have also sensory neurons that will then send information from the heart back to the brain. Now, what we also know is that when there is a miscommunication between these two organs, which is typically expressed as a sympathetic overdrive, there is a number of pathologies that they are really underlying this. So we see, for example, that we have sudden cardiac death that can happen under epilepsy. We have Takotsubo cardiomyopathy, where you have a contractile dysfunction due to sympathetic overdrive. And at the end, we also know that it is the sympathetic overdrive that is really leading to a heart failure and is driving this transition. So what we were interested in to study is what is really the communication and the interaction between neurons and cardiomyocytes. And to do this, we envisioned to do this in an organoid axis. So to generate a heart and brain axis in an organoid format generated by human iPSCs. So my group has been working for quite some time in bioengineering neuronal organoids. So we generate cortical organoids, motor neurons, and different kind of organoids. And of course, I'm embedded in the Institute of Pharmacology and Toxicology, where you heard, I think, enough also from Malte and Wolfram about the engineered human myocardium. Of course, we cannot connect the brain with the heart, or the brain organoid with a cardiac tissue. Therefore, what we are really missing is the sympathetic and the autonomic nervous system. And there was no other organoid out there, so we set out to generate one. So we did this with Lennart Schneider, a very talented PhD student. And to do this, we had actually to commit our iPSCs to trunk neural crest, where the sympathetic neurons derive from, and we had to manipulate a lot of different pathways in order to get the correct commitment. And what we are doing is we utilize what we knew from our brain organoids. So we knew that we can put iPSCs in the collagen hydrogel, and then we need to give them certain morphogenes. We need to give them a dual-SMART inhibition to make neurons, and now by utilizing different morphogenes, we can actually commit them to a trunk neural crest lineage. And you see here one of these organoids at day 6, and then at day 15 you see that the cells compacted now in the collagen, and at this stage we would expect FOX2B, FOX2A expression that would be neural crest markers, and later on we would expect that we would have noradrenergic enzyme markers like tyrosine hydroxylase and dopamine beta hydroxylase. So we started by really looking at the neural crest progenitors, and we compared our sympathetic neuronal organoid with our brain organoid, which we know wouldn't have a lot of peripheral neural crest progenitors, and that you can appreciate here in the whole organoid view as well as in the close-up. So we quantify this also by flow cytometry, and we could see that we have roughly around 60% of these progenitors, and we consider that this is quite a good amount for an organoid for the specific population we're looking at. And then we went a step further to see really about the noradrenergic, so the sympathetic neurons, and cholinergic, so parasympathetic neurons. We know that FOX2B progenitors would actually give rise to both. So we questioned what is the proportion really of sympathetic versus parasympathetic neurons. So we looked for markers by transcript expression, and you can see again, in comparison to our cortical model, we had a significant enrichment in the noradrenergic markers, so the sympathetic neurons. And we did have expression also of cholinergic neurons, but not so much higher than our brain organoid model. And when we look by immunofluorescence, then we can also see that we have a lot of cells that they are in the form of ganglia here, DBH positive, and you can see also the axons that they grow out of them. And last, we looked whether we had, of course, the neurotransmitters that we would require for the neurons to communicate with the cardiomyocytes. So we looked by LCMS, and we found that we actually had a significant enrichment of noradrenaline inside our organoids, where acetylcholine was present, but again, it was not really much more than the brain organoid. And if we see the levels, it looks like a 1-to-3 ratio. So we have now an organoid that has autonomic neurons, and we wanted to know, can we actually functionally connect cardiac and cardiomyocytes with neurons? So we did it in the simplest way I could imagine at the time. So since we have a ring, we decided to just try to push it and allow the organoid to fuse with the muscle. And we hypothesized that our neurons would actually then project towards the cardiomyocytes and will interact. So this is how our innovated organoid looks like at week 1. And if I show you here, you can see here is the muscle, and here in red you can see the neuronal organoid. And you can also appreciate that the tissue can continue to beat, so there is no steric inhibition by introducing our organoid in the middle. After 6 weeks of fusion, you can see that we have an immense innervation of the tissue. So you can see here neuronal axons towards the cardiomyocytes depicted here in red. And if we have also a close-up look, then we can also appreciate neuronal axons in very close proximity and interlacing the cardiomyocytes. And of course, we looked also for contractility. We did not want to make the tissues worse. So under our cell culture conditions, which of course are a bit different from what Malte is doing, because we also have a neuronal component that we need to maintain, we see that introducing actually the neurons and therefore having an innervated EHM, in this case we had an increase in cardiac contractility in comparison to the EHM. So how do we expect that our neurons look like in these tissues? We know not so many things actually from the literature, but there are some work from Zaglia and Monillo, which is beautiful, and they could show that we do have neurons that they are running around in between the cardiomyocytes, and then they show that there is a synaptic cleft, and that they release neurotransmitters that then can act on the cardiomyocytes. So of course, we wanted to look whether we have synaptic boutons that they run in close proximity to our tissue. And this is something that you can see here. We use synapsin, which is really showing you the synaptic boutons. And we also use tyrosine hydroxylase that is typically located in the synaptic boutons of the noradrenergic neurons. And what you can see here is a muscle in green, and then you see that you have neurons that they run in close proximity here with the muscle. And maybe you can see this a bit easier here, and you see synaptic boutons that indeed express and have tyrosine hydroxylase, and they are really on the cardiomyocytes. So, and I have also a video that it may run. So what we also found interesting was that we had a lot of neurons in the surface, and we did have neurons that of course went also in the deeper regions of the organoid, and We were interesting, however, because we have synaptic photons in regions where we don't have cardiomyocytes. So why would neurons form synapses, and with which cells? And another thing that we also observed was that many times in the tissue we seem to have indentations where we had cells that were in close proximity again with neurons, but these were not cardiomyocytes. And we saw that the muscle was actually growing around these indentations. So we coupled together with Dr. Fischer from the ZNI, and they did single-cell sequencing for us. This is only from 2 tissues, but I would really like to show you the data. So we found that we have different types of cardiomyocytes, as you can see here, and we have fibroblasts that we also expected because our tissues are generated also with fibroblasts, and obviously we have neurons, which is very good because we have a neuronal organoid. And we also found 2 interesting populations, chondrocytes and monocytes, a very small population, but very distinct. And what I found actually pretty interesting for our neurons is that we found a lot of vascular cells. So we have endothelial cells, we have pericytes, we have smooth muscle cells. And this is interesting because actually neurons are known to be attracted by pericytes and smooth muscles. So pericytes and smooth muscles secrete NGF, and this is what attracts neurons towards the heart during cardiac development. And of course, the first thing we had a look at is if NGF is really secreted in a specific population or generated in a specific population, and as you can see here, it is actually pericytes in our model that is generating NGF. So I wondered, do we really have the cells in a network? Do we really have just some endothelial cells and some pericytes not organized? So of course we did staining for these, and you could see here, this is the vascular network on the organoid, and we see here is the SNO, the sympathetic neuronal organoid, and here is of course the muscle. And we can also see that close to the PECAM positive cells, we have quite a lot of neurons, and probably that would explain the population that we see between the cardiomyocyte and the surface of the organoid. And this is a video again to show you. So what we also see is that the sympathetic neuronal organoid is giving a lot of cells that they are in the surface, and then we have quite a lot of vasculature, and at the end we see here the cardiomyocytes with a phalloidin. So it seems that the cells are actually cell sorting. So we have a population on the top, which I wouldn't dare to call it epithelium or epicardium, but they are all over the place, and we have in the subepicardiac layer quite a lot of vasculature and neurons, and then we see the neurons following the vasculature towards more the cardiomyocyte regions. These are very new data, but I was really excited 2 weeks ago, so I really wanted to show you. But of course, our main interest was, can we see the functional connectivity between the neurons and the cardiomyocytes? And for this, we decided to go in an optogenetic way. So we generated a line via CRISPR-Cas9. So that was done by another student, Kea Schmoll. And what she did is we used synapsin promoter so that we have it neuronal specific, and we used F-crimson, which is a redshifted variant of channel rhodopsin. And we generated this line, and of course then we made a sympathetic neuronal organoid, and we used multi-electrode array to really see if this organoid, what is the spontaneous activity, and also if we can really light stimulate it. And this you can see here, this is the basal activity of the organoid, so you see that our neurons are quite active, and you also see that we can increase this activity by light stimulation. And you can see here in the time course, of course, the weeks that this stimulation is having an effect. And next, of course, we wanted to see what happens if we actually fuse it with the muscle. And we hypothesized that if we stimulate the sympathetic neuronal organoid with a light, it would release noradrenaline, and then we will see an effect on the muscle. And that you can see in this video. So we have an EHM in the bottom, an IEHM, an innervated model in the top. and we will shine light. They both spontaneously beat, but only under light stimulation, the IEHM will increase its beating rate, whereas the EHM will keep a stable pace. And for us, this was extremely exciting. So you can imagine I was running in the labs extremely happy. But of course, we went and checked another 42 tissues to make sure that this happens, and we see that there is also a variability. And I think that this variability may be because whether the cells, whether the neurons will hit pacemakers or not, this is also right now a bit random. But we do see with all of the tissues, if you compare here with the post-stimulation, so no light, it is clear that we can stimulate by light all of these tissues. And we were also interested in a little bit in the parasympathetic part, and this will be more or less my last slide. So I just wanted to give you the last data. So we wanted to see what is really the parasympathetic component or the cholinergic component and what does it actually do. And therefore we decided to go with the chemical stimulation. So what we did is first we used atropine to completely block this effect and then see if really parasympathetic neurons are there, then we probably will have an increase of the beating rate. And what you can see here that indeed in the IEHM showed here in red, we have a small increase. It is statistically significant with the EHM, however, it's not significant with its basal activity before. So there is a small contribution, but not enormous. Then what we did is we gave nicotinic acid, and nicotinic acid would stimulate all the neurons, but since in the presence of atropine, we wouldn't be able to see the effect of acetylcholine, but only the effect on noradrenaline. And you can see here that again, we enhanced even further the beating rate of the tissues, and that shows that we have really, this is the pure noradrenergic effect. And last, of course, we blocked with propranolol to see if we can really abrogate this effect. And we can see here that when we repeat this whole experiment in the presence of propranolol, we don't see anymore a faster beating rate. So, and with this, I would like to conclude. I hope I could show you today that we generated a new model from iPSCs where we have innervated and vascularized human cardiac muscle tissue. We see that the cell distribution resembles really the heart, and it is an interesting cell sorting. And we know that our neurons and our cardiomyocytes are functionally interacting, and we are, of course, very interested now to go to disease and really look what happens if we have a disease model. And not only in comparing wild type with mutant tissues, the beauty of the tissue is that we are putting it together from two different sources, so we can actually mix and match. So we can use a healthy wild type line for the cardiomyocytes, and then a mutated neuron, and see what is the effect on the neuron. And we can also see really which is the cell type that is contributing then to the phenotype. And we set up a number of collaborations to look at different models. And of course, my dream would be at some day that we will have this axis. Now we kind of have all the tissues, but we really need to see on how to connect them all, because we know that, for example, Takotsubo syndrome, sudden cardiac death under epilepsy, we also need the central autonomic network. So with this, I would really like to thank the people that contributed to this work, Kea, Lennart, and Bao, and of course our collaborators, Wolfram for all the expertise, and Malte for the cardiac tissue engineering, our funding agencies, and of course you for your attention. Thank you.